I want to talk, shift gears a little bit and talk about IND prep versus commercial scale up, and then we'll get to some more audience questions as well. But we all know that, especially like in this novel protein therapeutic space, ADCs. Every conversation I have with an ADC manufacturer, at some point there's an in earnest discussion about speed to IND and efficiency to IND. But obviously, as we've ascertained, decisions made during that phase can create problems in upstream bioprocessing. So I want to throw the question out there. What decision points in early process development are most likely to become production issues when it's time to scale up for late clinical or commercial supply? And I think I'd start that question with Neeraj, if you don't mind, because you've seen some of these issues come to light.
Sure. Yeah, I think the specific to kind of getting ready for commercial production, let me kind of address that. I think from the standpoint of really, the drug supply to the market, FDA is heavily interested in, once you do get into the market, you want to be able to continuously, in an uninterrupted fashion, you want to supply. So kind of ensuring the supply risks and making sure that you are set up to produce and be able to supply to the market is the key. When I think about commercial production, that's what I think about. So with that said, I think there are two areas that you could really kind of think about it and potentially give that feedback to the early stage developers to say, well, here is what I need, right? So from the standpoint of, once you know the CQAs, once you know the attributes you're measuring, you want to be able to establish appropriate and streamlined commercial specifications so that you can produce the product in a consistent and an uninterrupted fashion. So that's the spec setting. Spec setting is a big area in itself that requires a very thoughtful process right from the get-go. How are you managing your batches? How are you going to set up your stability? And how are you going to bring all that experience and set a specification that you can justify before the regulators and kind of get the approvals? So that's one big area. And then the next one is capacity planning. When you are trying to produce, again, commercial production in an uninterrupted supply, do you have the right scales for a best case commercial demand versus a worst case demand, right? Commercial demand is never a solid number that you can rely on. There is always a big error bars, and as you know. And so therefore, you want to plan for those kind of things. What happens if the drug doesn't take off in a big way? What happens if it takes off in a big way, right? So you want to be kind of thinking about those extremes and say, well, my scale is appropriate, and then I have the capacity to produce, whether it is 20 batches or 40 batches, whatever it is, the case may be. So that's, I think, another big consideration that you want to be giving up. And the other big one, I think the last one I would say is that the shelf life. Again, you got to be planning for, in my experience, taking molecules into the commercial space, bare minimum, you want to get, you can get by with 18 months, but the 2 years is the starting point. So you want to be able to claim a 2-year shelf life and be able to justify before the regulators to, again, start your commercial production in a meaningful way. So I'll stop there with those 3 things.
You mentioned you mentioned manufacturability, and we did have a couple of questions. I know it feels like we're kind of moving on a continuum here and then taking a step back, but we had a couple of questions that relate to manufacturability that I wanted to get to. One from Lacan, and it's an important one. He asked what criteria is used to make the go no-go decision for novel or difficult to produce complex proteins, especially around manufacturability. And the second part of his question is, beyond potency, when do you decide it is not developable due to its complexity? Meinhard, I think I'd ask you to maybe start off with some thoughts on that.
Mm-hmm. Yeah, I think this is a good question. So what we do typically before we go into let's say, IND development, we perform a so-called late-stage developability study at the CDMO, where we produce the molecule at a certain already relevant scale with stable pool of clones, and we then typically apply certain process stresses like thermal stresses, shear stresses, and pH stresses, and other types of or light stresses to see how the molecule behaves and if it's robust or extremely sensitive. And out of such data, we would then, of course, decide if we can go already into a clinical development or in a clinical process development, or if we need some further discovery work to improve the molecule from a design perspective. And I think critical things would be here if there's a strong propensity to degradation in terms of protease degradation. This would be always hard to fix. So unless you have to add a lot of protease inhibitors during the whole process, manufacturing processes. And this is where I would see, let's say, the area where you have then to focus on. If you already have issues with manufacturing, that you really conduct a study to see the risk and then evaluate the risk. And eventually go back to molecular design.
Very well said. I think if I can put it in a little different way, I think what are the understanding the shelf life limiting factors, whether it is light sensitivity or some other mechanism, aggregation mechanism, whatever is the degradation mechanism that would really limit your shelf life. And again, I'll go back to the previous comment about if you feel like you're not going to get even a year of shelf life, I think that's going to be a huge, it's a no-go, I think, from my view standpoint. The other big one is the kind of connected, but also maybe not completely connected, is the productivity. If your titers and your productivity is so low, and you will have to go into 40,000 liter bioreactor to get to any kind of a decent amount of drug, I think then you got to pause. You got to pause and think about, well, is it going to be from a cost of goods standpoint? At the end of the day, the businesses are there to benefit from the molecule, and we are passionate about serving the patients, but the businesses need to survive themselves. So I think there is that cost benefit analysis need to be done as well. I think the shelf life goes into that. When you run the cost benefit analysis, the productivity goes into that factor. And so those would be some of the things you would think about in terms of go, no go decision.
Yeah. Lena, I want you to weigh in on this one too, because one, I see you nodding your head in agreement. I'm sure you've got things to add, given your perspective, seeing so many of these come across your plate. So
Yeah, definitely. I like this point, which Neeraj has also mentioned, the shelf life. So that is also the reason why we are often also recommending to do early formulation as well as product stability study. You can already do that also on the pool level. When you get product from the pool, cell pool, you can do pool production and then already start to have a look for formulation and product stability to be early, because you need to have a long time for stability, which you are testing to see which kind of stress factors are influencing finally the product stability. And that's a thing we would definitely recommend to do. And what I also want to add on is, definitely productivity. That is also finally when the thing when pharma companies or biotech companies are coming back to us. I saw that in the last year more often and often that companies are coming to us which have already a product in clinical study of clinical phase 2, and they want to go jump into the phase 3, and they realize, okay, our productivity is still too low for that kind of product, and we need to increase that to really make it a commercial, that there is good commercial availability finally, and enough money out of it finally, and that is what we also see. So in the first part, being good in the stability, being good in the productivity. So realize already is productivity enough what we are requiring, because you can also think about doing early more USP development to already improve productivity. That is definitely also possible, because we also saw a lot of companies who does not spend so much money into the USP part in that pace. But in my opinion, when putting on a little bit more effort into the USP, you often out of it and also improve not only the productivity but also the product quality finally. And that I would definitely add to Neeraj's comments here.